Good morning, Tribble students. So we've already looked at the way plants defend themselves. So right now we're going to be looking at how we can identify plant diseases and what we can do about them. So have a little think. How can you identify whether a plant has a disease or not? And if you can get to the extension, maybe define a disease. Maybe just pause, ignore that homework, that's from an old slide. Okay, so hopefully you gave it a little pause. I'm just thinking about, I've started gardening during this time, and I'm constantly just going out and looking at my plants to see if the leaves have changed colour. So that might be a really good way that you can tell whether a plant is diseased or not. So here's a really good example of discoloration of the leaves. We do find when they lose their green colour, it can be called chlorosis. And here we've got stunted growth, not growing properly. I've definitely got some of those that I've put in the wrong place in the garden and they've not grown as they should. A disease is a disorder of structure or function of a human, animal or plant that produces specific symptoms that affects a specific location and is not directly result to a physical injury. So generally a disease is normally, it's some kind of disorder or dysfunction in a specific location. Okay, so this is your list. It's going to be a very short lesson. You might want to go and Google some more stuff after this, but this is some short things. So stunted growth is caused by a nitrate deficiency. Now nitrates the plant gets from the soil. Remember that plants produce all of their own food via photosynthesis. But they do need minerals and ions from the ground, and they need nitrates. Now nitrates join on with glucose to make amino acids, which allows us to make protein, which allows plants to grow. So if you've got a plant with stunted growth, they probably need some fertilizer. And you probably remember when we did fertilizer about NPK fertilizers, so they're nitrate fertilizers. So this comes from Trilogy. So if we have spots on leaves, the chances are they've probably got black spot fungus. That's usually on roses, and that's the one that I said looked like it had chlorosis as well. And this is caused by a fungal infection, fungal spores. Black spots on roses or blights on potato are usually areas of decay. So bacterial fungi in the soil, particularly if it's not well draining soil, can cause blights. Growths, so like galls, can be caused by bacterial infections. I can never remember the name of the bacteria. It's like tuberreferens. You don't have to know the name, but bacterial infection causes galls or growths that sort of look a bit like tumours on plants. There we go, there's some. And then we've got malformed stems or leaves, and this could be because of an infestation of nematodes or aphids or something like that. So that's actually a pest. So you'd need to use an insecticide for that, really, wouldn't you? So like I said before, chlorosis is usually, or yellowing of the leaves is normally a sign of a magnesium deficiency. And magnesium is another ion that we will find in the soil. Magnesium is responsible for producing chlorophyll. So if you don't have any magnesium, you don't have any chlorophyll, therefore you can't absorb light, so you can't do photosynthesis. So magnesium deficiency is really quite serious. Discoloration can also be caused by tobacco mosaic virus. That's one of the pathogens you need to know for this course, and another link to Trilogy for you there. So discoloration can be caused by viruses, or by lack of magnesium. Other pests such as aphids and caterpillars, we already mentioned aphids. Aphids infestations can cause the stems to bend in certain ways, but other pests can also cause disease. Yeah, that one's not looking so hot there, is it? So they're winter moth caterpillars on the bottom left. Can't speak today. Okay, so sometimes students get a bit sidelined with certain diseases, certain questions in the exam. So here's a good example. You spot a disease plant in your paradise of a garden and you're going to do identify the disease. What would you do? How would you identify it and how would you treat it? Now, students aren't expected to know everything about all of them. If there are specific symptoms, then yes, you do need to know them. But perfectly fine answers are the following. Look it up on the internet or a gardening manual. That's a perfectly fine answer. Don't lose one mark for thinking, well, that seems too obvious. It's correct. Take the plant to a lab and identify the pathogen. Use a monoclonal antibody testing kit. We're going to talk about those in a second. You could analyse the DNA. That's another thing you can do. So if you look on the right here, it says monoclonal antibodies are identical antibodies produced by a single clone of a cell or cells. They only stick to one protein or pathogen, so it will give you a positive or negative test. So for example, monoclonal antibodies are used in pregnancy tests. So when a pregnant woman, normally have to wee on a stick, urinates on a pregnancy test, it will have monoclonal antibodies on there. And those monoclonal antibodies will stick to the hormone, the pregnancy hormone HCG, will stick to that pregnancy hormone and then change colour. And that's how you know when you're pregnant. Same thing for the AIDS test. So if you get tested for HIV, you will have your blood drawn. Your blood will go into a test which is full of antibodies that stick to the virus, the HIV virus, and that will change colour and give you a positive test. If it doesn't change colour, you haven't got that virus that it's sticking to. The test for this fungus can you see on the strawberry down there? You can test for that with monoclonal antibodies. Here's just a nice thing to finish up. So the sooner you treat something, the better. Then you don't have to cut down the entire plant or woodland or garden. There was recently, in the past couple of years, there was I believe it was a fungus or I think it was a fungus going through ash trees, and so many of them had to be chopped down because it carried really quickly. So let's quickly pause this, link them up. Let's go. So botrytis. How will we treat these? Well, we would use an antifungal treatment because it is a fungus. Black spot, same, it's a fungus. Aphids, pesticides, because aphids are a pest. Nitrate deficiency, well, we're going to have to whack in some fertilizer. Insects are pesticides. Magnesium is in fertilizers. Tobacco mosaic virus, generally we need to remove the plant because it spreads really fast, and same for bacterial infections. So those are the main things you need to worry about plants. I'd say nitrates and magnesium deficiencies are the ones often forgotten. So please do make a really good note of that. Okay, so we have got some exam questions to have a go at. So this is the tobacco mosaic virus. It's infected there. All tools should be washed and disinfected after the plants are infected. Suggest why. So these are just 2 marks. It says we'll obviously swap by, we'll just pause. And scientists produced a single plant with a resistant gene. How can they use it to produce many plants? So this is linking back to cloning. So we would wash all of our tools to prevent the virus from spreading. And we would take a cutting or a tissue culture, or we'd take stem cells from the meristems, and then we would produce clones. Just to remind us, if you've forgotten, the meristem is where we find stem cells or undifferentiated cells, which means that the meristem cells can grow into a whole new plant. They can grow into any organ of a plant. Really, really clever. Here's another couple of questions. Some plants produce fruits which contain glucose. Describe how you could test for the presence of glucose. So this is food testing from when you did digestion. And TMV can cause plants to produce less chlorophyll. This causes discoloration. Explain why plants with TMV have stunted growth. So just another one to show you. This is all just linking back to our photosynthesis stuff. Have a pause, see if you can answer these questions. Benedict solution will turn from blue to brick red in the presence of glucose. And if you've got less chlorophyll, you have less photosynthesis, less glucose is produced, less energy, so less growth, less proteins, less cellulose, all of those things. Jot those down because people tend to forget all of the uses of glucose produced by photosynthesis.